Welcome students. Today we are going to study the applications of ultraviolet spectroscopy. Ultraviolet spectroscopy has been mainly applied for the detection of functional groups, chromophore, the extent of conjugation, detection of polynuclear compounds by comparison, etc. Well, there are n number of applications of UV spectroscopy. So here we'll be only discussing some important applications. Well, you can study many more apart from these. So today we are starting. First application is the detection of functional groups. The UV spectroscopy is applied to detect the presence or absence of the chromophore. The absence of a band at a particular wavelength may be regarded as an evidence for the absence of a particular group in the compound. If the molecule is very complicated, then only little information can be drawn from its UV spectrum. Well, the spectrum of a molecule is transparent above 200 millimicron, then it shows the absence of first conjugation, second a carbonyl group aldehydes and ketones, third benzene or aromatic compounds, and fourth bromo or iodo atoms. An isolated double bond or some other atoms or groups may be present in the molecule. It means that no definite conclusion can be drawn if the molecule absorbs below 200 millimicron. Then second is identification of an unknown compound. An unknown compound can be identified by comparing its spectrum with the spectrum of known compounds. Now, if the spectrum of both the compounds, that is known and unknown compound, they coincide, then we can say that two compounds are identical. But if the UV spectra of both the compounds, that is unknown and known compounds, they do not coincide, then the expected structure is different from the known compound. That means the unknown compound is not the same as the known compound. So it's a different compound. So we can identify an unknown compound by UV spectroscopy. Then next comes the extent of conjugation. The extent of conjugation in polyenes R CH double bond CH whole N R can be estimated. Adhesion in unsaturation with the increase in the number of double bonds, that is increase in the value of N, shifts the absorption to longer wavelength. And it is found that the absorption occurs in the visible region, that is at about 420 millimicron if N is equal to 8 in the above polyene. Such an alkene will appear colored to our human eye. We can see it colored. The next is distinction in conjugated and non-conjugated compounds. UV spectroscopy also distinguishes between a conjugated and a non-conjugated compound. The following isomers can be readily distinguished since one is conjugated and the other is not. So here the structure of both the isomers are written, and you can see the first one is conjugated and the second one is non-conjugated. So the forbidden N to pi antibonding band for carbonyl group in the compound first will appear at longer wavelength compared to that for the compound second, which is non-conjugated. The alkyl substitution in an alkene causes a bathochromic shift, but this technique is not much useful for the identification of individual alkenes. Then next is the examination of polynuclear hydrocarbons. Polynuclear hydrocarbons and benzene, they have characteristic spectrum in the UV and visible region. Thus, the identification of the polynuclear hydrocarbons can be made by comparison with the spectra of known polynuclear compounds. The presence of substituents on the ring generally shifts the absorption maximum to longer wavelengths. Then the next applications of UV spectroscopy is preference over two dotomeric forms. If a molecule exists in two dotomeric forms, preference of one over the other can be detected by UV spectroscopy. Now here you can see two structures, that is 2-hydroxypyridine and pyridone 2. So 2-hydroxypyridine exists in equilibrium with its tautomeric form pyridone 2. So the spectra of these two compounds were found to favor pyridone 2, which is an alpha beta unsaturated ketone, and clearly the equilibrium shifts towards the right, that is pyridone 2. So by the help of UV spectroscopy, if a molecule exists in two tautomeric forms, preference of one over the other can be detected. The next one is identification of a compound in different solvents. Sometimes the structure of the compound changes with the change in the solvent. Chloral hydrate shows an absorption maximum at 290 millimicron in hexane, while the absorption disappears in the aqueous solution. So that means the compound contains a carbonyl group, that is aldehyde, in hexane solution, and its structure is CCl3 CHOH2O, whereas in aqueous solution it is present as CCl3 CHOH whole twice. Well, there are many more other applications also. For example, let us study one more, determination of configuration of geometrical isomers. The results of absorption shows that cis alkenes absorb at different wavelengths as compared to their corresponding trans isomers. The distinction becomes possible when one of the isomers is forced to be non-coplanar by steric hindrance. Thus, cis form suffer distortion and absorption occurs at lower wavelength. Well, if we consider the spectra of cis and trans stilbenes, so we'll say that for cis stilbene, the lambda max is 280 millimicron, and for trans stilbene, the lambda max is 295.5 millimicron. So here we can see the cis form suffers distortion and absorption occurs at lower wavelength. So here lambda max is 280 millimicron only. So the examination, one more is elucidation of the structure of vitamin K can also be done by the help of UV spectroscopy, and there are many other applications also. So these were the main applications which we discussed today. Thank you so much.